Assalamu alaikum friends. I hope that you all will be fine. Friends, in this video I'm going to discuss about the last topic from protein metabolism, and that is glycine metabolism. Okay. We will cover this topic under the headings of sources, the breakdown of glycine, the formation of beneficial products from the metabolism of glycine, and disorder. Okay. So let's start our lecture with the sources of the glycine. Okay. Sources. So the first source of the glycine is serine. This serine, it forms glycine by the action of serine hydroxy methyl transferase enzyme. Okay. So this is the first source of the glycine, that serine, it forms glycine by the action of serine hydroxy methyl transferase enzyme. Now coming toward the second source, threonine. This threonine also, it forms glycine by the action of an enzyme, and that is threonine aldolase enzyme. Okay. So these are the sources of the glycine, and must remember that serine and threonine both are hydroxyl group containing amino acid. This serine forms glycine by action of serine hydroxy methyl transferase enzyme, and threonine, it forms glycine by the action of threonine aldolase enzyme. Another one source of glycine is ammonia, carbon dioxide, and 5 and 10 methylene tetrahydrofolate. They also form glycine with the help of glycine synthase enzyme. Okay, so friends, these are the three sources of the glycine: serine, threonine, and ammonia, carbon dioxide, and 5 and 10 methylene tetrahydrofolate. These are the three sources of the glycine. Now coming toward the degradation of glycine. The degradation of glycine. Okay, so first of all, this glycine, by action of oxidase enzyme, it forms glyoxylate. This glycine, by action of this oxidase enzyme, glycine oxidase enzyme, it forms glyoxylate. This glyoxylate, by action of transaminase enzyme, it forms this glycine. So this is the basically the degradation of the glycine, and must remember that this glyoxylate, it also breaks down into oxalate and formate. Okay, this oxalate, it gets excreted through our kidney, this gets excreted through urine, and this formate, it is very important in one carbon metabolism. So this is basically the degradation of glycine, that glycine, by action of glycine oxidase enzyme, it forms glyoxylate. By action of transaminase enzyme, it forms again glycine. This glyoxylate, it is very important. It breaks down into this oxalate and formate, and this formate is very important in one carbon metabolism. And this oxalate, it gets excreted through the urine. Okay, so friends, now coming toward the formation of the beneficial product by glycine metabolism. Formation of beneficial product. Okay, so first of all, this glycine, it plays a very important role in synthesis of heme. This glycine basically, it gets condensed with succinyl CoA, and through multiple reaction it forms heme. So this is the first product from the glycine metabolism, that this glycine gets condensed with succinyl CoA and through multiple reactions it forms heme. Another one, this glycine, it also gets attached with arginine and it forms guanidoacetate in kidney. Okay, it forms guanidoacetate in kidney. Basically, this glycine, it gets attached, it gets condensed with arginine and it forms guanidoacetate in kidney. In next reaction, this guanidoacetate, by action of methyltransferase enzyme, and here S-adenosyl methionine, it forms S-adenosyl homocysteine, and there is conversion of guanidoacetate into creatine. And this creatine, in next reaction, by action of creatine kinase enzyme, it forms creatine phosphate. In next reaction, by phosphatase enzyme, and phosphatase enzyme is responsible for the removal of phosphate from this substrate. By action of phosphatase enzyme, it forms creatinine. Okay, so these are also beneficial products. These are also products for the metabolism of glycine. That glycine, it gets attached with arginine and it forms guanidoacetate in kidney, and by action of methyltransferase enzyme it forms creatine, this product. And by action of creatine kinase enzyme, kinase is responsible for phosphorylation, addition of phosphate group in substrate, it forms creatine phosphate. And by action of this phosphatase enzyme, this enzyme is responsible for removal of phosphate group from substrate, it forms creatinine. Another one, that this in conjugation reaction, this glycine also involves in conjugation reaction. Basically, this glycine, it gets condensed with benzoyl CoA and it forms hippuric acid, and this hippuric acid, it gets excreted through our kidney. So these are the formation of beneficial products. And another one is glutathione. The full form of glutathione is gamma glutamyl cysteine glycine. Okay, and also this glycine also plays a very important role in the formation of glycocolate and glycogenodeoxycholate, that are bile salts, and these bile salts are very important in the digestion of lipid. So basically these are the formation of beneficial products from the glycine metabolism. That first one is heme, another one is creatine and creatinine, and another one is hippuric acid that is excreted through our urine, and glutathione, full form of glutathione is gamma glutamyl cysteine glycine, and last one is the synthesis of the glycocolate and glycogenodeoxycolate. So these are the formation of beneficial products. And now coming toward the disorder of the glycine metabolism. Disorders. So disorder basically the deficiency of transaminase enzyme, it leads to disorder, that is deficiency of transaminase enzyme. It leads to disorder, and that is primary hyper hyperoxaluria. So this is the disorder. The deficiency of this transaminase enzyme, it leads to disorder, that is primary hyperoxaluria. Now another one is, this is the first one disorder. Another disorder is that glycinuria. It is due to the defect in absorption of glycine through renal tubules. Okay, that leads to excretion of glycine in urine. So these are disorder, the deficiency of transaminase enzyme, it leads to disorder, that is primary hyperoxaluria, and another one is glycinuria, which is due to defect in the absorption of glycine through renal tubules and leads to excretion